Hey, Mr. Reed, I have a question. Will you come over here and answer it?
Of course I will, young student. Because after all, I'm Mr. Reed.
Hooray!
What is your question?
I was wondering how babies are able to be born.
Well, that is a good question. And I will try to answer it as clearly as I can. First, you must understand the biology behind it. Babies are made when a human male gamete and a human female gamete come together and fertilize.
What is a gamete?
Gametes are the result of a cellular process known as meiosis.
I have never heard of meiosis before either.
No worry, I shall teach you all about the stages and outcomes of meiosis.
Cool.
Yes, it is. Now listen up. Meiosis occurs by a series of steps that resemble the steps of mitosis. Two major phases of meiosis occur, meiosis I and meiosis II. During meiosis I, a single cell divides into two. During meiosis II, those two cells both divide again. The same demarcating phases of mitosis take place in meiosis I and meiosis II. Would you like me to clarify what the phases are?
Sure.
At the beginning of meiosis I, a human cell contains 46 chromosomes, or 92 chromatides, which is the same number as during mitosis. Meiosis I proceeds through these following phases. The first is prophase I. Prophase I is similar in some ways to prophase in mitosis. The chromatides shorten and thicken, and become visible under a microscope. These are known as the homologous chromosomes, and in this case, there are two. The two homologous chromosomes come near each other. Because each homologous chromosome consists of two chromatides, there are actually four chromatides aligned next to one another. This combination of four chromatides is called a tetrade, and the coming together is called synapsis. After synapsis has taken place, the process of crossing over occurs. In this process, segments of DNA from one chromatide in the tetrade pass to another chromatide in the tetrade. These exchanges of chromosomal segments occur in a complex and poorly understood manner. They result in a genetically new chromatide. After crossing over has taken place, the four chromatides of the tetrade are genetically different from the original four chromatides. Now on to the next phase, which is metaphase I. In metaphase I of meiosis, the tetrads align on the equatorial plate, as they also do in mitosis. Centromers then attach to spindle fibers, which extend from the poles of the cell. One centromer attaches per spindle fiber. After that we go to the next phase, called anaphase I. In anaphase I, the homologous chromosomes separate. One homologous chromosome, consisting of two chromatides, moves to one side of the cell, while the other homologous chromosome, also consisting of two chromatides, moves to the other side of the cell. The result is that 23 chromosomes move to one side, and 23 move to the other. Essentially, the chromosome number of the cell is halved. For this reason, the process is a reduction division. Now for the final stage of meiosis I, which is telophase I. In telophase I of meiosis, the nucleus reorganizes, the chromosomes become chromatin, and a cytoplasmic division into the two cells takes place. This process occurs differently in plant and animal cells, just as in mitosis. The two daughter cells, both with 23 chromosomes, then enter into phase, during which there is no duplication of the DNA. The interphase period may be brief or very long, depending on the species of organism. And with that, we are finished with meiosis I.
Yes. That is great.
Now on to meiosis II. Meiosis II is the second major subdivision of meiosis. It occurs in essentially the same way as mitosis. In meiosis II, A cell containing 46 chromatids undergoes division into 2 cells, each with 23 chromosomes. Meiosis II proceeds through the following phases. It starts with prophase II. In prophase II, the chromatin material condenses, and each chromosome contains 2 chromatid attached by the centromer. The 23 chromatid pairs then move to the equatorial plate. The next phase is metaphase II. In metaphase II, the 23 chromatid pairs gather at the center of the cell prior to separation. This process is identical to metaphase in meiosis. The phase after this is anaphase II. In anaphase II, the centromers divide, and the 46 chromatids become known as the 46 chromosomes. These chromosomes then separate from one another. Spindle fibers move one chromosome from each pair to one end of the cell and the other member of the other pair to the other side. In all, 23 chromosomes move to each side. And finally, we are at the last phase of meiosis II. This last phase is telophase II, during which the chromosomes gather at the sides of the cells and become indistinct. They then form a massive chromatin. The nucleus envelope develops, then the nucleoli reappear, and finally the cells undergo cytokinesis as in meiosis. And that's all for the process of meiosis. Now, would you like me to explain the process of meiosis? No big deal, I'll just go watch videos of cows mating.
Oh, I might as well.